Thank you very much for inviting me to this interesting meeting. I'm going to talk to you about something that is very close to my heart, which is how to measure aging. And I have no conflict of interest, not whatsoever. I'll start by showing you a number of individuals in the wide age range. And I selected successful individuals on purpose because I'll show you that even in people that maintain strong cognitive and physical function, the effect of aging are still very, very evident. And so that is to make the point that measuring aging should be possible, but it's not as simple as it may look like at first sight. We usually talk about older people by focusing on these individuals, the individuals that are very, very old. But in reality, the aging process affects the individual across the entire lifespan. And measuring it across the entire lifespan becomes extremely important because maybe we could be able to capture those individuals that age faster than others early in time when intervention that are supposed to slow down aging are still possible. So how do we measure aging? This is the slide that conceptualized the way aging occurs in my mind. So you can see that there are two circles. One is talking about damage, the red one. The green one is talking about resilient reserve. And you can think about aging as a constant battle between these two forces, the accumulation of stochastic damage of molecules, cells, and organs, and then a number of very complex and wonderful evolutionary selected mechanisms that both avoid this damage or repair this structure when the damage is done. And when the resilient reserve is no longer able to maintain the damage low, then the individual becomes frail. But there are other two ways where the individual can have accelerated aging. The first one is that it is exposed to something that is environmental or intrinsic, that is highly damaging and overcome the capacity of the resilience mechanism. Or the resilience mechanism may be lower to start with, either because they have been damaged early in life or because of genetic disposition. And this individual will have early mortality and early frailty in their life. But how do we measure this? How do we translate this concept into experimental design? And to me, it's very important that to do that, we understand that there are several metrics of aging. These are the three that I follow in designing my studies. And of course, what is the most important one is functional aging. The dream of every older person is to get to old age able to enjoy everything they can do in life and have cognitively intact capacity. But these functions are really affected by what we call the phenotype, the body composition, the ability to create energy, the brain health. And underneath these mechanisms, there are biological mechanisms, what we call the biological mechanisms of aging, that ensure that there is integrity so that there is no or there is low decline of physical function. There are many studies that have tried and attempted to obtain measures of the biological process of aging. This is my friend Steve Horvath that developed an epigenetic clock. The CpG methylation sites are methylated or unmethylated in the millions in our genome. But he selected 353 of them and created a weighted average of them. And this weighted average truly track aging beautifully. But on top of that, not only track aging, because we could measure aging by a clock, deviation, individuals that deviate from this average trajectory of methylation aging tend to have adverse outcome. For example, this is another epigenetic clock that was done by Mork and Levine, it's called PhenoAge, suggesting that the deviation for normal aging from the methylation perspective is associated with mortality. And there are many other studies that show that slow epigenetic aging is associated with things that tend to be good for your body: moderate alcohol consumption, fish consumption, exercise, and high socioeconomic status, while faster epigenetic aging is associated with bad things: high blood pressure, inflammation, atherosclerosis, and central obesity. Other people in our group, for example, have used circulating protein. And for example, here we use circulating protein, 1500 circulating protein, to create a proteomic clock. As you can see, track aging beautifully. And even in this case, deviation, those individuals that deviate from this longitude, this trajectory, tend to have adverse outcomes. And so the problem with these metrics that started to be used in some of the clinical trials, that many of these metrics were calculated based on cross-sectional data. And cross-sectional data are highly problematic. For example, if I want to compare people between the age of 20 to 100, I can think that their difference is due to their different age. But in fact, most of the difference is due to previous experience. This individual at age 50 was 20 at the time of the Green Revolution. This individual 90 was 20 at the age of Great Depression. And this individual under 5 was truly age 20 in World War I. And clearly, this experience has shaped the biology and the susceptibility of this individual. The second issue is that cross-sectional studies eliminate the people who die or don't come back to follow up because they become sick. And for example, if you look at the number of disease in the Baltimore longitudinal study of aging, there is a certain rate of increase with aging, but when you look at the longitudinal cohort, the rate of increase is much, much steeper. And there is a problem in calculating metrics of aging with cross-sectional data. And for example, we can say that an individual that deviates from the normal trajectory may have been already older biologically to start with, do not experience accelerated aging. And there is all sort of combination of primary genetic environmental effect that we cannot calculate from cross-sectional data. Instead, some other studies have used a longitudinal trajectory of some phenotype that are reported here. This is from Dawn Belsky and Terry Moffitt, showing that if you sum up phenotype that change with aging, you can have a much more powerful rate of aging metric. Then you can use that to calculate and as a reference to calculate a much more powerful epigenetic clock. And that really has been shown by demonstrating that the longitudinal derived metrics are much more predictive of bad outcome than those that were calculated cross-sectionally. One of the problems with this approach is that by calculating that, you don't really take into account that many of the characteristics of aging do not change linearly, but change curvilinearly with aging. For example, if you take fat mass, more men and women tend to increase with aging, but then above the age of 70, there is a substantial decline. So two individuals that are shown here will have accelerated aging if you compare them with this time, but if you compare them with those that are in their 90, their trajectory will be normal. So we need to compare individuals based on the trajectory that are measured in the general population. And that's exactly what we have done. If you look, for example, at this idealistic phenotype, you see that these are the individual, the thin line, and these are the population trajectory. And these are how much this single individual that are the black dot deviate from the population trajectory that is curvilinear. These are the rate of change of this phenotype. In this case, what was the specific phenotype derived from energetics. And so if you sum up their distance from the average phenotype, you obtain, and I don't know why it doesn't advance. Well, first of all, I'll show you that you need to take into account nonlinearity, because many of these phenotypes change nonlinearly with aging, both in energetic and body composition, and so on. And then when you sum up this distance from the normal trajectory of aging across different phenotypes, you obtain distribution that show that there is large heterogeneity both in young and old age, although at old age the heterogeneity is large, and deviation in this heterogeneity are associated with bad outcome. Here, for example, is a predictive of cognitive function and physical function, such as memory and time to finish 400-meter walk. And the age equivalent difference of one standard deviation of this score really predict individual to be younger, for example, almost 14 here on average in visual spatial capacity. And why even more important, you see that these metrics work beautifully at any age, both in young, middle age, and old age, suggesting that this variability is predictive of outcome over time, even at young age, will allow us to really capture individuals with accelerated aging that are target for prevention and intervention. And in the cognitive domain, that is exactly similar to what I show you in the physical domain. I'll stop here to say that it will be nice if something made sense for a change. I think that, something that seems so simple as measuring aging end up being very complex. And there are many researchers that are working on this issue. And first of all, it would be impossible to do all this work alone. And so I need to recognize the great work that is done in these studies by my team. Thank you very much.